Bismillah ar-Rahman ar-Rahim. Assalamu alaikum, dear students. Welcome to the online class. I'm here, your course instructor Muhammad Waqas for the course Environmental Microbiology ENS-224. In today's lecture, we will talk about the activities of soil microorganisms. Before studying the role of microorganism, first of all, we will talk about energy and nutritional flow in the ecosystem. That is the producer, the role of producer, the role of consumer, and the role of a decomposer. After that, we will talk about the role of microbes in the metabolism of organic and inorganic compounds. But for today, we will talk about the role of microbes in the organic metabolisms. And among the organic compound, the first one we will talk about the carbon cycle. And among the carbon cycle, we will discuss about the cellulose degradation, the lignin degradation, and the synthesis of humus. The first one, if you talk about the energy and nutritional flow in ecosystem, as you know, all living things must obtain energy and nutrients from the abiotic and biotic environment. This energy and nutritional relationship in ecosystem can be described in a number of convenient ways. For example, the food chain or energy pyramid provide a simple summary of the general trophic levels, or simply we can say the feeding levels, right, designated as producer, consumer, and decomposers, and traces the flow and quality of the available energy from one level to another level, means from the producer to the consumer and the decomposers. And it's worth nothing that the microbes are the only living things that exist at all three major trophic levels, means the microbes can be producers, the microbes can be the consumers, and the microbes can be the decomposers. The first one is the producers. Life would not be possible without producers because they provide the fundamental energy source that drive the trophic pyramid. Producers are only organisms in ecosystem that can produce organic carbon compounds such as glucose by fixing inorganic carbon like carbon dioxide from the atmosphere. And such organisms can be termed as the autotrophs. Most producers are autotrophs, or simply we can say most producers are the photosynthetic organisms such as plants and cyanobacteria that convert the sun energy into energy of chemical bonds. See, in addition to these, a small but important amount of carbon dioxide assimilation is brought about by an unusual bacteria that is called lithotrophs. We have discussed the lithotrophs in the introductory classes, that how the lithotrophs work, right? The metabolism of these organisms derives energy from the oxidation reduction, or simply we can say the redox reactions of simple inorganic compounds like sulfides and hydrogen. In certain ecosystem, the lithotrophs are the sole supporters of the energy pyramids. The second one is the consumers. Consumers feed on other living organisms and obtain energy from bonds present in the organic substrate that they contain. The category includes the animals, the protozoa, and few bacteria and fungal species. The pyramid usually have several levels from the consumers, ranging from the primary consumers, which consume the producer, to secondary consumer, that is the carnivores, which feed on primary consumers, to the tertiary consumer which feed on the secondary consumer, and up to the quaternary consumer, usually the last level, which feed on the tertiary consumer. As you can see in this figure, the primary consumers, the paramecium, of that is secondary consumer, tertiary, and the top carnivores. The third one is the decomposers. Primary microbes inhabiting soil and water break down and absorb the organic matter of dead organisms, including plants, animals, and other microbes. Because of their biological functions, the decomposers are active at all levels of food pyramids. And it's thought that without this important nutritional clause of saprobes, simply we can say saprophetics characteristics, the biosphere would stagnate and would die. The work of decomposers is to reduce organic matter into inorganic minerals and gases that can be cycled back into the ecosystem, especially for the use of primary producer. And this cycling back of organic matter into inorganic minerals and gases is termed as the mineralization. It's so efficient that almost all biological products can be reduced by some type of decomposers. For decomposers, numerous bacterial species decompose cellulose and lignin, which are complex polysaccharide from plant cell walls that account for the vast bulk of detritus soil and water. Both fungi and bacteria decompose such complex animal compounds such as keratin and chitins. The second one is the role of microbes in the metabolism of organic and inorganic compound. The first one is the role of soil microbes in the metabolism of organic compounds. As you know, microbes produce and transform the organic matter by producing biomass of their own cell. As I told you that microbes are not working for us. Basically, they are trying for themselves to need to meet their needs. So in simply we can say microbes produce and transform the organic matter by producing biomass of their own cells and collect substrates essential for recycling and the supplies of humus. They also decompose and mineralize the organic compound and consequently recirculating the indispensable elements in plant production based on the assimilation of carbon dioxide from atmosphere. The first one among the organic metabolism is the carbon cycle. As you know that 50% of the organic matter is made up of carbon, and that carbon gets back into the soil in the form of plant and animal residues, in the form of, for example, falling of leaves, various remains in meadows and forests, animals, roots and shoots of dead plants. The fresh organic matter is composed of monosaccharides like hexose, pentose, polysaccharides like starch, cellulose, organic acids, aromatic compounds like lignin, phenols, and hydrophobic compounds like wax, fats, etc. The carbon recovery is done during the decomposition and mineralization of organic compound. However, the speed of decomposition and mineralization of the carbon depend on the chemical nature and particular component of substrate mass. For instance, the soluble substances such as sugar, amino acid, organic acids are easily washed away with water from the residues of plant and animal and then are quickly metabolized by the soil microbes. What is the result? They especially regulate the microbiological activities in the rhizosphere. Similarly, for the complex compound like waxes, fats, rubbers are decomposed with great difficulty due to their high hydrophobic properties. For instance, among the plant material, lignin is the most resistant to decompose. The first one is the cellulose decomposition. Cellulose is basically a carbon-based compound. Cellulose is a polysaccharide that consists of long unbound chain of glucose units. They occur commonly in the cell wall of plants and is associated with hemicellulose and lignin. In the dry mass of green plants, the content of cellulose is ranged from 15 to 30%, whereas in the lignified parts and straw, it's contribute up to 50%. And if we talk about its decomposition, the best known cellulitic system occurs in fungi, meaning that fungi are the only microbes that can betterly degrade the cellulose. The trichoderma genera release the most active cellulose enzymes into the environment, that is followed by the enzymatic attack on the cells. Similarly, the bacteria that degrade the cellulose belong to genera Cytophaga and Silvibro species. Furthermore, the decomposition may also occur in the oxygen-free condition. Mostly the fungi and the cellulotic bacteria degrade the cellulose in the presence of oxygen. Then simply we can say during aerobic conditions. So here it means that the cellulose can also be degraded and during the anaerobic condition. The decomposition may also occur in oxygen-free condition, that is conducted by the genera Acetobacter species, Bacteroides, Clostridium species, and consequently after the degradation, a large number of gaseous substances like carbon dioxide, hydrogen, and methane are created. For the degradation in the soil, the decomposition of cellulose occurs fastly in the soil with the pH of neutral and slightly acidic pH, whereas in highly acidic soils, the decomposition rate is a bit little slow, or simply we can say in comparative to neutral and slightly acidic soils, the decomposition rate in the highly acidic soil is slow. The second one is the lignin degradation. As you know, lignin is also a carbon-based material. Lignin belongs to a large group of aromatic compounds, and apart from cellulose, it's the main component of tissues, you can say wood tissue, that's contribute up to 30% of the plant biomass. Lignin is also basically a polymer that is built of phenylpropane units which contain an aromatic rings with methoxyl group, that is OCH3 group, right? The most active lignin degradation organisms are the fungi, that so we can say wide rot of wood. They decompose wood, right, to carbon dioxide and water. And these group of fungi belongs to Basidiomycete and Ascomycetes groups and are represented by several hundred species. Similarly, among the mold fungi, the Trichoderma species also demonstrate the ability to decompose lignin. For the degradation of lignin, basically there are two different mechanisms, the direct degradation and the indirect degradation. The direct decomposition takes place by leachase that may oxidize the monophenols and lignin peroxidase. There's also enzyme and some other enzyme that directly degrade the lignin into simpler component, right? Simply we can say to carbon dioxide and water. Similarly, in the indirect decomposition, the enzymatic decomposes lignin is represented by what? The oxidoreductase, which require oxygen or hydrogen peroxide for the oxidative tearing up of bonds that connect the phenylpropane subunits of lignin to each other, right? And the activity of microbes that decompose the lignin in soil stimulates the production of the humic substances. The third one is the synthesis of humic substance, or simply we can say the humus synthesis. What is humus? Humus is an organic substance, usually dark in color, that makes up a system of large surface area. If a substance is the high surface area, it is capable of absorbing different type of ions from water and gases. And the process of humus formation, or simply we can say the process of humus formation, is termed as humification. The composition of humus is the fulvic, humic acid, and humans. These are the corporation basically of more or less carbonized compound, which is characterized by the presence of different groups such as carboxyl group, phenyl group, methoxyl group that contain carbon, oxygen, nitrogen, phosphorus, and sulfur, as well as some aromatic skeleton with numerous side chains. However, the main substrate from which the humus compound are formed are lignin, hydrocarbons, and nitrogen compounds. For human farming system, the main humus forming system is the activity of soil microbes like bacteria, including actinomycetes and fungi. Basically, what they do, these microbes conduct different type of processes connected to the formation of humus. What are these processes? First of all, they degrade the fresh organic matter by resulting various metabolites, and these metabolites are a precursor of humic compound. Secondly, the microbe, what do they create? Different type of substances which after atrophy and autolysis. First of all, what is atrophy? Basically, atrophy is the progressive disintegration, especially as a result of degradation, or simply we can say the degeneration of cells. And what is autolysis? Auto means self. The destruction of cells or tissue by their own enzymes. Basically, what these enzymes come from, especially those enzymes are released by the cell lysosomes. So, the microbes creates the substances which after autotrophy and autolysis make up additional initial substrate that is needed for the formation of humus. Thirdly, the microbes catalyze the process of human synthesis, means they speed up the formation of humic reactions. And if we talk about the basic pathways of the humus synthesis, that is, if we talk about the stage number 1, the basic way of humic compound formation is through its synthesis from the fragments, that is polyphenols, with the participation of nitrogen components of protein origin. Mainly, proteins are the rich source of nitrogen. So, the source of polyphenols may be the processes of lignin decomposition, hydrocarbon transformation, and various other microbial synthesis processes. Similarly, many polyphenols form as a result of metabolites of different microbes. The second stage after the synthesis is the humification. Is the oxidation of polyphenols, right? That has catalyzed by the phenol oxidases. These are the enzymes produced by different microbes, such as by fungus, circular species. And in the final stage is the polymerization process, right? So the final phase of the process is the polymerization of the oxidized phenol. So the 3 stages. The first one stage is the synthesis, right? After that, the second stage is the polyphenol oxidation, and the third stage is the polymerization of the polyphenol oxidation. So the transformation of all these of the organic matter in condition when oxygen is available. So it means that these stages or all these processes are carried out in the presence of oxygen. So the transformation of the organic matter in condition when oxygen is available leads to the formation of humus. For example, in the absence of oxygen, or simply we can say during the anaerobic oxygen, it leads to the formation of peat deposition, or simply we can say during anaerobic condition, it leads to the peat formation. And this is all about for today class. And inshallah, in next lecture, we will talk about the role of microbes in the nitrogen cycle. Thank you so much for today class. See you in next class.